Hi, guys. Can you hear me okay?
Yep.
Did anybody go and eat some Fig Newtons this weekend? I thought I'd bring my happy Easter tree in today for all of you to enjoy. Little bunnies, little chicks. Jason, way to go. Way to be a trooper, testing it out. All those delicious wasps. Hey, everybody. So we got an email over the weekend to make sure that we checked in on everybody being active and still being able to see what's going on and participate. So I sent out those emails this morning just to make sure we didn't lose anybody. And I also sent out an email with our community homework. So I'll talk a little bit about that in a little bit. But I did promise you, this might be the one time you see it. I'm drinking my tea with my senior mug. See how I've retained my youthful vivaciousness? Oh my God, they're horrible. They were gifts to my grandparents, and I couldn't throw them away because I know they loved them so much. But anyway, cheers, cheers to everyone. Class of 1994, Mansfield High School, Massachusetts. All right. So hope you guys all had a great weekend. As I mentioned, that we got a couple of blasts of messages, and I just wanted to make sure there's somebody who's logged on in the past as Dr. Worm. I don't know who that person is. So if anybody knows what that screen name is, I just want to make sure I don't mark anybody for not like being active because they're Dr. Worm and I just don't know who Dr. Worm is. Thank you, my tree. This was a little like self-care that I gave myself last week after I felt a little reprieve of no stress. So thank you for those of you that are enjoying the Easter tree. Okay, so I'm gonna share the screen so we can get started. Are you guys seeing a big PowerPoint?
Yep.
Okay. All right. So we left off with symbiosis. And remember, symbiosis was a tightly linked association of two different organisms, and it can be positive, neutral, or negative. The negative is the parasitism, which we covered. The neutral was commensalism. There are not a lot of examples of that, but we did talk about like barnacles on other shellfish and on, you know, whale fins. We mentioned, I think, those burrs on animals and so on, and epiphytes as well. And then we talked a little bit about mutualism. We were kind of ending off with mutualism. So I want to pick up today. Did anybody have any questions on what we covered so far? Okay, if you do, put a chat on. I'm hearing the door. They're breaking in. Are you all right, Rowan? Sorry, guys. So we talked about things like, you know, once a parasite attacks an organism, you know, what are the responses? Hold on one second. I can't help you. I'm on my class. I can't help you set it up. There's a request to set up Minecraft, except I don't know how to do it on that computer. I'm sorry, guys. Anyways, for some reason. Okay, we talked about terms. I just want to make sure you guys are comfortable with these terms, right? Micro and macro, big and small, macro being the big, micro being the small. Remember that. The macro will have multiple hosts maybe because they're bigger. Over the course of their lifespan, they might be molting into different life stages. We talked endo and ecto, and that was timely with the virus being the endo inside and the ecto being a little larger. Again, fleas, ticks, that sort of thing. We talked about COVID a little bit, really important for us to be informed about that stuff. We mentioned another endoparasite being the measles virus with the hotspot of resurgence due to some of those anti-vaccination areas. We mentioned Lyme disease being complex, involving both an endoparasite, which would be the spirochete bacteria that's picked up when a tick, a black-legged tick, also known as the deer tick, infects, or actually, I'm sorry, feeds on a mouse where they pick up that bacteria. Then they go through their life stages, feeding on other hosts, transmitting that. So the endoparasite is going to be our Borrelia burgdorferi, and our ectoparasite is going to be the tick vector. And we mentioned that, you know, we can't think of the disease without the greater context of the community. And, you know, I've had a lot of time to reflect more recently about what I want you guys to get out of the class. And what I really want you to get out of the class, which normally would be like some field experiences and Excel and, you know, that sort of thing. But in general, I think I want you to walk away this semester understanding the complexity and the interactions between organisms and abiotic factors like climate, rain, and temperature changes, and then the things that we do on the landscape. So, you know, our role as an organism interacting with these plants and animals, and how that all shapes what's going on. So the example given here is with, you know, Lyme disease, and that you tend to have more Lyme disease where you have more disturbance and less large forest tracts. Smaller forest has more edge, more scrubby habitat where you'll find a lot of paramyscus, and those paramyscus are the mice that will carry these diseases. Hold on one second. Rowan, can you please lower that a little bit? I have to go lower that. You guys are probably hearing Minecraft noise. Hold on. All right, we also mentioned some terms like holo and hemiparasite. And a holoparasite is a complete parasite. A hemi is partial. These are the plant parasite terms. The holo meaning complete, this is going to be something that is not going to photosynthesize on its own. So it'll be mostly kind of white and has to tap in to its host in order to survive. But a hemi is going to be able to photosynthesize when it's not necessarily with an ideal host. So it will have its own ability to photosynthesize. If you did a cross section of mistletoe, you would see that it can do that as well. And when an organism gets parasitized, right, you're going to want to not immediately die if you are the host that it's attacking, right? You're going to need to have some sort of evolutionary strategy to reduce that. So oftentimes it's behavioral. You can shift the location where you're going to be located to maybe be in an area where there's fewer of those parasites, or you could groom them off or have a helper groom them off. We also mentioned that plants can be parasitized. So that's where this bullet gall kind of came into play. You guys remember, for those of you that were on, right? I have this oak bullet gall. And so a parasitic organism, the females laying her larvae, they are developing in that. That is a response of the plant that's being parasitized to grow some sort of like scab type thing so that it won't infect the rest of the plant. Eventually that animal will emerge, and we've seen some of those emergence holes in a lot of the things that I've picked up in the woods around here. But again, it's a way, just like with humans, when they get attacked, to have a response, some kind of an immune response. Our tissues respond, and here the plant tissues are responding. We gave some examples about how a parasite can regulate, right? Any organism that can alter birth and death rates or both on a host would be considered a regulatory organism, right? And so we see that here with the parasite that attacks these ants. It turns their gaster abdomen area into something red, looks like a berry. Birds jump in, eat it, defecate elsewhere, and you'll see those parasites kind of continue through their cycle that way. And there was a lot of good videos embedded and hyperlinked in. If you guys missed some, make sure you guys go back and check them out. We mentioned, again, regulation, altering births. Here's an example, two examples, right? Any parasitized lizard tends to have fewer babies. Any parasitized zebrafinch or bird that might actually have secondary sex characteristics that makes them bright, they'll get fewer matings if they're not as bright, not eating as good food because they've been parasitized. And in addition, the females can't seem to hang on to the fat, and therefore they don't have as many babies. In terms of mortality, we're looking at deaths, and the examples here. We've got the rabbit, when it's infected with tularemia, is going to act a little slower than usual and get attacked more often, probably by raptors or coyote. And then for aquatic organisms, anything that gets parasitized might swim differently, school differently. Their behavior is going to attract the attention of predators like osprey, and they can knock their numbers down. And you'll see that behavior just increase, you know, even more so when you have more parasite load. We mentioned the isopods, the pill bugs. So if any of you guys lifted up any of those rocks and seen these little cool pill bugs, their response is to kind of curl up. I play with my kids and I kind of go on a little hunt for these guys, but you'll notice that, you know, their strategy to avoid being eaten is to hide under cover. And anytime they are parasitized, they will not hide as often and become more susceptible to predation. So if they're not parasitized, they are hiding under cover, and if they are not. We mentioned, you know, what happens with parasites. What is this cycle? We know that they have to weigh out whether they're going to be super strong and virulent and overpower their hosts. And that's kind of weighed out with how they transmit across the population. And so they're always trying to balance that, almost like an optimal foraging type thing. And so you can see this SIR model where you've got a lot of susceptibles in the beginning. As those taper down, because now some are becoming infected, we see that peak, and then when that starts to decline, that's where we're picking up those recovered organisms. And again, that's the whole idea with flattening the curve. You know, if we can stretch out the number of infected by social distancing, we can keep it such that we're not going to overwhelm the healthcare system, which is what we're all kind of hoping for. And one thing I did want to ask, and let me know if you guys are in need. I don't know if there's a way that I can help with this, but if you had to go grocery shopping and stuff, did the school or does the dorm provide you guys with any kind of like gloves, rubber gloves? Anybody? I just wonder if they could leave some in the dorms or in an area where you could access them before you went out grocery shopping. It makes me a little worried.
Yeah, my twin is grocery shopping right now. Should he have worn gloves?
So I haven't been wearing my N95 mask, and I have a few of them because I do my small mammal trapping. But I think if, as long as I'm social distanced, and I have my gloves, you know, they have the sanitary wipes that you can wipe the carts with. And then when I get home, I've got my gloves on the whole time. I take them off when I get in the car to drive home. And then I put some on to like carry the bags in. And then I've been like crazy mom wiping everything down with bleach wipes, the Clorox wipes, which you
Yeah.
probably make on your own. Also, there's a lot of recipes out there for homemade hand sanitizer. If you can't find it in the stores, it involves rubbing alcohol and aloe vera. And if you have essential oils and you want it to smell nice. But I would definitely recommend it if you can access some sort of gloves or just wear something that, even if they're winter gloves, I would think that's maybe better than nothing that can be washed afterwards. But perhaps reaching out to the health center and seeing if there is that access for you guys. So keep me posted. I don't want anyone risking it. Definitely washing your hands, and having hand sanitizer in the car before you touch your steering wheel and all that stuff would be very, very important. I know we all got that warning about that crazy person, which hopefully wasn't one of you guys, that went out and infected, or was out in the general public knowing that they had tested positive. Super, super important to just use our common sense. But anyway, if you need anything, if you think that I might be able to reach somebody, I can inquire about that. I worry about you guys taking unnecessary risks. Now, kind of switching back, poverty and these parasites we tend to see in lower socioeconomic areas, more encounters with these parasites. And there's, again, a lot of stuff in the news about parasites. Now, parasitism is like predation, right? It was a plus-minus interaction. Somebody benefits, somebody is negatively impacted. Commensalism is a +0. So that negative, that harmed individual has sort of developed some way to kind of reduce their harm. And that's an evolutionary strategy. So these were all the examples that we had given and I had mentioned earlier. And mutualisms were the plus with everybody benefiting, and we were kind of we're finishing up on this note. Everybody's happy. It's a symbiotic relationship where each of them are gaining some sort of a benefit, whether it's they're growing better, surviving better, having more babies. But again, circumstances, environmental things can change, and the reason why you might need that helper might go away. And therefore, you might see the relationship change as the environment changes. So that's something to consider, that it may not always be a beneficial thing. We do know that these organisms can increase. If you have a helper, you can increase your carrying capacity, which is pretty awesome. And we did also learn that anytime you're in a stressful habitat, like say the desert or the tundra, you may see more community interactions that are mutualistic, where it would be beneficial to have a helper. And in kind of more reasonable, less stressful environments, you might see more competitive interactions. So that's kind of a trend that we tend to see more often. But again, that can change, right? It's context dependent, right? So when soil nitrogen, soil phosphorus are low, it makes sense to have helpers, whether they are bacteria that can fix this nutrient for you and give it to the plant, or have more fungus associated with your root hairs to pick up some of the water and those trace nutrients. That would be really useful. But if all of a sudden somebody fertilizes that area, then there's no point in having those helpers. They can actually end up harming you because they're still trying to tap in and grab your carbon if you're a plant. And again, if you have a mutualism that involves having an insect that will, be on your plant, and like an ant, fighting off aggressively other insects that want to eat your leaves, if all of a sudden you don't have those other insects that want to eat your leaves, then why would you want to have these ants on there that you have to feed with fat? So again, context dependency. The cleaneress, that was another example, right? So we have these tropical fish, they have these helpers that are off kind of eating the ectoparasites on the outside. But if they get too aggressive, if their numbers get too much, they might actually end up eating some of that protective mucus layer, which can be pretty bad. And so you can see that it actually benefits in terms of community diversity. You have much more diversity in the amount of fish in these coral reef areas when you have these cleaner fish, because it reduces parasite load. Again, sometimes it can go the other direction when the numbers get too high. We talked about partnerships in a mutualistic relationship, right? Is it required? Is it not required? If it's required, it's obligate, it's obligated, they have to be together. And if it's not, then it's considered facultative. So the examples we gave with the yucca and the yucca moth, okay, here's that yucca again, right? So you can see the beautiful white flowers. And the moth, there's a unique moth for each. Those flowers will turn into a fruit, and the fruit actually has a hard shell, and I think you guys are seeing the hole, right? Where that moth is going to emerge. Danny is laughing, so I'm assuming he's seeing it. Or maybe he's chatting with somebody else, I don't know. But anyways, it's important to note a good example of an obligate type of a relationship is the yucca moth, and also the fig wasp. Those would be good examples. And again, there's that arms race, right? So the moth wants to, the female wants to lay her larvae in wants to lay the larvae in that flower fruit, combo. And the plant wants them to kind of roll around in the pollen and transmit it across, other individuals. So we get cross-pollination. But if that moth female lays way too many of those babies, then we will see that plant drop the flowers and fruit. That is their sort of strategy. If they've taken too much, if that parasite's taken too much, mutualist is taken too much and acting like a parasite, that plant will respond and drop its flower, its fruit, okay? And then we saw I brought them just in case I was hungry, right? The fig Newtons. Delish, right? We know that we've got internal flowers of these figs. We've got these fig wasps, and the female's tunneling in, and she's gonna lay her larvae inside these flowers, gonna lay a gall. Well, it's gonna form a gall type structure. They'll emerge. They'll be looking around. The males will be looking around for somebody to mate with. The males will dig out these tunnels. Boys, I am on a lecture. Boys, quiet. And then they will emerge. Females will go into another flower, and the cycle kind of begins again, and some of them get left behind. So I had talked about how they might crunch a little bit, maybe some exoskeletons. So coevolution, these mutualisms, other really good examples of obligates would be, look at the proboscis, look at that kind of tongue mouth part on this moth, and the orchid that it pollinates. You guys have probably seen the Darwin orchid and the Sphinx moth. Obviously co-evolved from one another. The plant is going to be pollinated, and the moth is going to gain some nectar reward, right? Co-evolve from one another. You lose one, you lose them both. And then we've got another one. We don't have termites up here yet, I should say, but in the south, termites are a big deal. Up here, we have carpenter ants, right? So what we've got going on here in the termite gut is having helpers, just like we saw with, cows and their rumen, how they have bacteria, protozoa that will help to digest things that we can't and get those nutrients out. So in this case, we have a lot of helpers in the guts here, and they're gaining the nutrients they need, and these guys are gaining their nutrients. Facultative mutualism. So mycorrhizal fungi are an example of this. And it's a fungus that basically is just extending the size of the root hairs, right? You can kind of see, they call that the mycosphere, right? So we're talking about increasing your surface area for absorption of water and other trace nutrients, right? The plant's going to grow better. But they're not necessarily required to survive. But imagine like a really harsh location, some maybe country where you're trying to grow crops and the soil is really, really poor. Then if you can inoculate the plant, the crop that you're trying to use, with a mycorrhizal fungi, you might be able to get that plant to grow and increase yield, which would be a win-win all around the board. Other types of mutualisms, there's 3 types over here. Pollination, we've given examples with the yucca moth and the fig wasp, and the moth. the orchid, right? So those are all kind of obvious. The pollinator gets the nectar reward, the plant gets pollinated. There's other ones like defensive and cleaning. So we've already alluded to the defensive with the ant and acacia story. And so in a situation where, Rowan, can you not fall on top of the computer? Can you back away? Can you back away? Don't jump on the bed. Sorry, we had to reconfigure so that we can have two people in this like office bedroom. He's up on the bed jumping right now. So anyways, if you have a lot of herbivores around, say like kind of leaf cutting insects, if you have ants on the acacia, they will fight those other insects that want to eat the plant. And if you don't, you will have a way more of those insects that will eat the plant tissue. So anytime you got a lot of herbivores around, it makes sense for the acacia plant to have a lot of those ants. What will it do for the ants? It's going to give them, like I said, a fat reward in their belching body. So those big thorns on the acacia right here, they're hollow, gives them a place to stay, but they also have access to high nutrient kind of fatty material that the plant exudes for them. And so that's their reward. And the plant doesn't get increased herbivory. Boys, I need you to be quiet if you're staying in here. And the cleaning, well, actually, there's another defensive, right? So there's some grass that actually has fungus inside of them. The fungal hyphae is actually very distasteful to grazing animals. So imagine you have a lot of cows around, and then the cows, so there's Rowan, and then the cows don't want to eat it because it gives them kind of an upset stomach. So that's one kind of, thing that a farmer could do. They could plant a crop that has that engineered in them to reduce herbivory. And the cleaning, we've got the wrasse story that we talked about, but the oxpecker is another bird that might actually eat some of the ectoparasites off of cows and cattle and other organisms. In terms of other mutualisms, ants are really important in so many systems. I'm not sure if you guys are aware of this, but some plants, I believe Trillium, if you guys have ever been out on a walk in the springtime, Trillium has 3 leaves. It's a beautiful little flower, and it grows in these kind of clonal cluster areas. But the seeds of those Trillium have these little eliosomes, and they're protein and fat. They're really like prized by any kind of insect. So that is one strategy. The plant seed doesn't want to stay underneath its parent, right? If it does, it has to compete with its parent. So it really makes sense for them to have either some structure that like spirals them away as the wind blows them. Think about the Pinocchio nose, on the maple trees, those samaras that I used to put on my nose as a kid. Maybe you guys did, I don't know, maybe I'm crazy. But that helps them spiral away. The dandelions, when they poof in the wind, that helps as well. But these eliosomes will be like a high draw for the ants, which will bring them back far away from the parent plant to their burrows. And they may actually germinate there, giving them a chance to germinate. Notice, whoops, the number of seeds is really high right near the parent plant, right? This is distance from the parent plant. The farther away, there's fewer of them, but the probability of germinating and surviving, the farther away you get from your parent plant, because you're not competing, is higher. So that is an important strategy. Bloodroot and Trillium, really important strategies so that they can increase their population sizes. Again, the cleaning example, right? But this is with sharks. Pilot fish will eat the parasites on the outside of the sharks. And it can be commensal given, whether or not the numbers are different. Other common classic examples, these are ones you should just kind of commit to memory, would be nitrogen fixing in legumes. So if any of you guys have had, beans, obviously, alfalfa, clover, think about cover crops. So when corn is grown, usually the next year farmers will plant something different. And so the plants that they will plant that next year tend to be nitrogen fixing plants, meaning that they have the ability to take up rhizobia, which are these bacteria in the soil, and create these root nodules. You guys see those clusters on the root hairs over there. Those root nodules are kind of like galls, right? The rhizobia bacteria will go inside those root nodules, and the bacteria has the ability to take atmospheric nitrogen and convert it to a nitrogen form that the plant can use, because plants can't use atmospheric N2 gas. They just can't. And that's like the most nitrogen we have of all, right? So it's important for them to convert it to, say, like ammonium or nitrate or nitrite, that the plants can uptake it. And so the help of that bacteria will do that. Well, what's good? Why is that important for the bacteria? The bacteria need sugars, right? They need carbon. So they're going to tap into the plant's roots to get the carbon from the plant, and they're trading off the nitrogen that they can't have, the plant can't have on their own. Lichen, another example. A lichen is, whoops. A lichen is going to be a plant or photosynthetic organism, cyanobacteria type thing, and a fungus, right? So the fungus surrounds that photosynthetic partner, giving them kind of moisture and protection from UV sunlight and desiccation. And in turn, since it's a fungus and can't make its own food, it's going to be getting sugars from that photosynthetic partner. And so again, the photosynthetic partner is not drying out getting too hot, and then the fungus is getting its carbon. And that's how that lichen works. And we've seen them on trees. You've probably seen them on gravestones, seen them on the ground, on rocks as well. A recently discovered mutualism, which I think is totally awesome, is with salamanders. And this may be true in some other amphibians as well, but they seem to have gain the ability to pull in some algae into their eggs. And as a result, they're gaining oxygen. It's kind of, the question is still out, what is the algae gaining in this, right? It's clear what the salamander is gaining, but they think maybe it's gaining a unique habitat. But again, research is still kind of ongoing for that. I just thought that was pretty cool, especially since a lot of the animals are in the vernal pools right now, getting ready to emerge. And our final example of a mutualism is with marsh grass. So remember, the marsh is where the muskrats are located, mostly herbaceous wetland. And the grasses actually provide a little bit of shade, the right microhabitat for these mussels to grow at their roots, right at the roots, at the kind of interface there. And so the mussels actually, because they have those strong byssal threads, like imagine the ones on the rocky intertidal, it can stabilize the soil in case there's like storm surge or erosion, high water, flooding, that sort of thing. So the plants are gaining that, and the mussels are also altering salinity, which is also kind of important, and the plants and so on. So back and forth, there's kind of an abiotic trade-off with these guys. So again, what is a symbiosis? It's a tight relationship between two different organisms. It can be plus minus, it can be plus zero, it can be plus plus. We talked about types of parasites: endo, ecto, micro, macro, holo, hemi, right, for the plants. We mentioned some examples of how they can regulate their population, altering birth and death. We mentioned commensalism with some examples. We characterized our mutualisms. We talked about obligates and facultative as the partners, with lots of examples of all of these different types. Okay, any questions on mutualism, parasitism, commensalism? Good? Okay, again, lots of good videos there if you guys wanted to check them out if you missed them last time. So the lecture, I'm hoping you guys are you seeing this new lecture? Okay, all right, so we're moving on to, Rowan, please get down. You're gonna fall. We're moving on to communities, right? We have left our populations with population growth. We started talking about the community interactions, and now we're going to talk about how do we quantify these communities. So this ties nicely with our spider lab, where you guys were looking at species richness and the Simpson index, as well as the Jacquard community beta index. Rowan, it's flipping. Get off. Sorry, he's climbing up the exercise bike. So what I have pictured here are different communities, rocky intertidal, we've got a meadow, we have an alpine, well, it's actually a bahada. If any of you guys have been to Tucson, Mount Lemmon is a pretty cool drive. It actually can take you like up biomes, but in a vertical direction. So rather than like driving from Arizona on up to, say, Oregon, you could actually drive Mount Lemmon and see those biomes kind of switch a little bit. So there's alpine stuff at the summit, and there's kind of deserty stuff down at the bottom. It's really cool if you get out to Arizona. So we're going to define what a community is, which I think we already have in the past, but you know, imagine a cookie cutter that you just dropped out of an airplane, a giant cookie cutter, and it encompasses plants and animals and every other organism you can imagine in the same space and time. That is a community. We're going to talk about how we measure communities, and again, it ties in with lab. Rank abundance graphs are important to note. Very easy to interpret two different communities and which one is more species rich and even, just with a quick glance at this graph. So I think they're really informative. We'll talk about how community structure, having a lot of habitat heterogeneity, and complexity, you know, like low canopy, mid canopy, high canopy. Think of tropical rainforest. There's a lot of structure, right? So you can imagine that community is going to be very diverse, rather than maybe a monoculture where there's not many. We talked about keystones already, but we'll bring it up again for reinforcement. That's an important community structuring organism. And then we'll talk about how we measure between community diversity. So the beta diversity, like with Jacquard and the Sorensen index. And then we'll talk about these two old fogies, Clements and Gleason. They are our community ecologists. They both kind of had these ideas as to what was important in structuring a community. Is it the abiotic factors and each species' individual life history? That's very much an individualistic Gleasonian concept. Or do we think of communities as sort of interacting, kind of the clockmaker thing? Every organism has such a tight interaction with another that you can't really think of a community without thinking them all interacting together like a clock. And that's a Clemensonian kind of an idea. Okay, so again, by definition, a community, it's a group of different species, plants, animals, microorganisms, all in the same space and time. And we look at their diversity. Diversity is good, right? The more diverse animals that you've got in your community, the less disease you will have, at least with Lyme disease in many cases. It's a buffer, you know, it suggests there's a healthy system. So alpha diversity, we talked about that in lab, and beta diversity as well. Alpha diversity is the diversity we see within one community, and beta is the diversity we see between two communities. So the beta would be like the Jacquard index, where you compared, you know, island 1 to 2 and 2 to 3 and 1 to 3 and so on. Whereas if you were just looking at species richness in community 1, or just looking at the Simpson index for community 1, that would be an example of alpha diversity. So you can see in this picture over here, I have 4 different sites, kind of like our 4 different spider islands, right? And the first thing that we might want to do to look at alpha diversity is just, what is the species richness of each of these communities? And again, that's the easiest measure of diversity of all, okay? And it's not the N number of individuals. It's the number of species we see. So let's take a look at site 1, right? We have 1, 2, 3, 4, 5 different representing species here, right? But we only have 1 of each of them, right? So that does mean that our species richness is 5, okay? Not only do we have 1 representative of each, but we have 5 different species. Species richness is 5. Over here in site 2, we have fewer species, right? So our species richness is less. We have 1 that's represented twice, right? The pine tree. And then we have another one that's represented twice, right? This deciduous tree in full leaf. And then we have 1 over here by itself. So 1, 2, 3. Okay, so that's where the 3 comes in. And over here again 5, and over here we have 3, right? So hopefully you guys, that's familiar. It's you can understand it. We're not looking at number of individuals. We're looking at the number of different unique species. That's species richness. or big S, okay? That is the best, easiest, quickest measure of diversity in a community. The next thing that we can do is look at relative abundance. Remember P sub I in the spider lab? That was the number N individuals of that one particular species divided by total in your whole sample, okay? That's P sub I. Evenness kind of breaks out your relative abundance and looks at them and says, are all of the species relative abundances about the same, or do we have some that are really high and some that are really low? If some are really high and some are really low in that community, that usually means that we have a dominant species in the community and then everybody else is rare. If all of them are about even P sub I, or relative abundance, then we have, you know, equitability or evenness in that community, which can be good, right? You don't want to have, you know, one dominant species, then all of a sudden a tsunami comes in and you lose it. There's going to be a trophic cascade in that community, right? Functional diversity, again, is there structure in our I'm sorry, that's the physical structure. Functional diversity is what's the role that organism plays. So if I'm a pollinator, but I can be a generalist pollinator and you know, pollinate everything, not just you know, low elevation plants, I can do high elevation plants, then that means I can serve, you know, multiple functions in this community. If I lose those high elevation pollinators, but I could still go up there, then we'll retain the function of pollination in that community. And so thinking about niche is where that plays in. Okay, so let's take a look at an example. This is very similar to the homework that I sent you guys in an email. The first two sections of the homework are you guys going through and filling out 3 blank columns. The first blank column is going to be your relative abundance, or P sub I. Your next one is going to be your ln, your natural log of that value, ln of P sub I. And then your final column will be a cross multiplication, kind of like we did Lx times Mx. It'll be your P sub I value times your ln of P sub I value. And you'll see that in a sec, okay? So this is a good example for us to walk through. Again, we're looking at a tree community right now. This is one tree community that's very, very diverse, right? There's over 20 species of trees in this one community in northern West Virginia, okay? And what we did is sort of like what you'll be doing in lab this week, right? We go out, we make a 10-meter square plot in the woods, and then we count up the number of individual trees of each particular species and get a count. Okay, so in this case, there were 76 yellow poplar, there was 36 white oaks, 17 black oaks. And when we add all the N number of trees that we counted, we get 256 of them, okay, as total. So your P sub I is just the N for that particular species divided by your total N, okay? So the column I have right next to this is the N column, right? The counts. The column I have right next to that is really your P sub I column, right? The relative abundance. It is your N for that particular species, in this case 76, divided by your total 256. The next one is 36 divided by 256. 17 divided by 256. I prefer you guys to leave it as a decimal. Don't scale it up to like percent, right? Don't multiply it by 100 to make it a percent. These guys made it a percent, but I would rather you leave it as like 0.297, 0.141, okay? For the purposes of what we're doing. So all of them should add up to 1 if we leave it as a decimal in terms of the P sub I's, or to the 100% if you had scaled them up to 100%, okay? Now this table is ordered so that the first couple that you see are the ones that are most common in the system, right? And that'll be important a little later on. Is everybody okay with this table? Okay, we're just doing relative abundance. N for that species divided by total. All right, we'll go over to another community. Now we're gonna compare. That was community A, this is community B. Let's compare them, okay? And in this case, we have much fewer species, so species richness should be less, right? And we had 122 yellow poplar compared to, say, 76 of the yellow poplar, okay? We had 274 individuals total Okay, we had 256 in the other one. And again, to do P sub I, the column right next to the ends, it's going to be one, you know, the n value divided by your total, 107 divided by total, 12 divided by total. And don't leave them as a percent, you know, keep them as the decimal as I had asked before, okay? So that should be pretty straightforward. Again, when you open your homework up, you're gonna see two tables for question like, or part 1 and 2. And the first thing I give you is your species column, then I give you your n, number of individuals column. Your next column will be you doing that P sub I, relative abundance, n divided by total. Okay, so sum all of the n's up, get your total, and then that'll be your denominator on all of those P sub i's. All right, so that rank abundance graph, I want to make sure we get to this. That rank abundance graph is pretty cool because the communities that we just looked at, the yellow one was the community we just saw. Okay, you can look at this graph and you can see where the line ends. Okay, the line ends at 10. Okay, the line here ends at 24. What does that mean? That means this rank abundance on the x-axis. That means that I can look at this graph and I can say, you know what? Community 2 has a species richness of 10. The length of that line is species richness, okay? Community 1 that we looked at has a species richness of 24, okay? So anytime you see a rank abundance graph, look at the length of it. Whatever one is longer will have a community that has the greatest species richness. Okay, that's important to note. The other thing that we can gain from this rank abundance graph is look at the slope of it, okay? Our y-axis is relative abundance, or P sub I, or the log of P sub I, depending on the scale we're looking at. The flatter the line, the more even the community is, right? It would mean that my P sub i's, for the most part, on almost all of them, are about the same. My relative abundance, my proportion of each of them, is about the same. And that would suggest the flatter your line, whoops, the more even your community is. The steeper your line suggests that you probably have like 1 or 2 of them that are the most dominant, and then everybody else is not represented very well. They're very rare, okay? So any steep line that you have would be a not even, uneven community that's dominated by just a couple, and there's a few rare. And then anything that seems a little bit, the slope is more gradual in general, would be a more even community, which again, for diversity, for disease, reducing disease, the more even your community, the better, right? Because oftentimes the ones that dominate are like your invasive species or like mice, right? So that's really important. And the reason why we don't throw like species names down here is this ranking is unique for each of those communities. So ranking number 1 is going to be whatever the most common species, it's associated with whatever the most common species is in your table. In this case, both of them were most dominated by yellow poplar, okay? But your number here that's ranked 2, notice it's sassafras in this community, it's it's white oak in the other community. So rather than put species names in here, you just rank them according to, you know, what species is the most dominant. It doesn't matter what species in particular. It's just looking at the trend, okay? And so that's important to note. And you can see here, this is a community, and just a kind of hypothetical. Both have the same species richness, 5, right? Community B is much more even. Community A is dominated by 1 species and everybody else is rare. This is lizards, real life stuff, right? A eucalyptus plantation versus a forest where we find the same species, more complexity, right? There's way more species richness in the forest than in the monoculture plantation, and it's a little more even, right? We would say it's more even though there's a big, you know, there's a couple dominant, right? In general, most of the represented species are more evenly distributed than in the monoculture, which is less complex, okay? Getting to the homework, okay, we've walked through this a little bit. This is what your homework looks like. You've got your species names here in one column, you've got your N values in the next column, you've got your P sub i's, which we just talked about how to do that, right? It should sum up to 1 or 100%. Just your N 84 divided by 256 is this one. 4 divided by 256 is this one, right? Your n divided by your total. The next column is your ln of p sub I. So just hit your ln button, okay, on the calculator, the natural log. It's going to give you a negative value. Don't sweat it. Leave the negatives there, okay? Your final column is pi times ln pi. Very straightforward. Take your pi column and multiply it by your ln pi column. So this cell times this cell gives you this. This cell times this cell gives you this, okay? Now, if we're looking to see how diverse your community is, and you've just filled out your table, all you need to do, kind of like we did with R0, right, is sum the whole final column up and take the negative of it. So like absolute value it. Okay, so add all of these up, pretend almost like they don't have a negative on them. Okay, you add it all up, you get this negative 1.3661. Get rid of the negative sign, like you take an absolute value. And that H value right there is the Shannon-Wiener diversity index, okay? Kind of like our Simpson index, it's a measure of diversity in that community. And so the example, you know, the calculation is here. H is the negative sum, so absolute value, right, of the PI times the ln PI column. So simply fill this column out, sum it up, get rid of your negative sign, and that is your H. What does that mean? Diversity ranges from 0 to 1, okay? I'm sorry, diversity ranges from 0 to an H maximum, okay? So you guys also have to figure out what your H maximum is. So if you know what your species richness is, in this case there's 5 species, H max is the ln of your species richness, okay? So that is another question I ask on the homework. What's your H max for that table? Figure out how many species you've got and take the ln of that. Okay, so if there's 5 species, it's the ln of 5. If there's 7 species, it's the ln of 7. That is your H max. So your diversity ranges from the 0 to whatever your H max is in that community. If it's closer to H max, you've got a diverse community. If it's closer to 0, you're not a diverse community. You're probably dominated by a couple species and you've got some rarity, okay? Now, how do we figure out evenness? Evenness is another question I ask. It's straightforward, okay? Just take the H you just calculated from summing the, you know, and taking the negative of that column and divide it by the H max you just calculated. So H divided by H max is your evenness. We call it J, okay? And that value is down here for this particular table, okay? And so in terms of the homework, that gets you through most of the homework. I'm going to skip forward. We have a lot of time on our hands for our next lecture, but I'm going to skip forward to the final, flip the homework over to the back page. There's just this calculation left to do. It's very much like the Jacquard index, okay? And it's very quick for you to see. If you don't want to mark your animals, you don't want to tag them, you don't want to paint them, then it's good enough to have, say, a game camera in one location and say, you know what, we got cheetah and ocelot and we got house cat. That's a species richness of 3 in that rainforest, okay? And then over here, you just got like panda, dog, and that's it, okay? And so now we have species richness in 2 communities. We don't have N values. We didn't count them. We didn't mark them. Some people don't like to tag animals, right? But we have species richness in each of those communities. So we can do, just like we did with Jacquard, notice I have the Jacquard down here. How many of those species are in common? That's the little c, divided by how many of those species are shared. So add the, you know, what's the total of all the species with the 2 communities combined? That's the denominator. That's Jacquard's similarity index. Again, this is a beta. We're comparing between the 2 communities. For Sorensen, it's kind of similar, okay? It's 2 times the number of species in common. In the example I just gave you, if I'm remembering, we didn't have any of species in common, right? So that would be 0. And then the denominator is the sum of your species richnesses in those 2 communities. The first community I gave you, I think, had 3 cats. The second community I gave you had like 2 animals, right? The panda and the dog. So 3 plus 2 is 5. And so you would divide 0, no, none in common, divided by 5. So that would be a 0 for the Sorensen similarity index. I give the example in the next slide, right? So it's just presence absence data. What's my species richness? If you know species richness of 2 communities, you can compare them. It's 2 times the number of species that they have in common divided by the sum of their species richnesses. They range from 0 to 1, okay? And if there's a 0, if it ends up being 0, like the example I just gave you, they have no species in common. Those communities are totally unique, very different. And if it's close to 1 or closer to 1, or anything above like a 0.55, you know, or above a 0.5, right? You can say that the community is more similar, but you could also say, you know, there's like 10% more species in this other community than this one. But anything close to 1, you would say these are almost identical communities. They share species. Anything close to 0, they're totally unique communities. Nothing is shared. Okay, so that will get you guys through the homework. Again, it's not due this week. If you can do it by the end of the week, that would be great. Our quiz is scheduled for the 8th. It'll be an online Moodle thing, you know, obviously not many questions. It'll probably embed, you know, a little bit of this math to make sure you're comfortable with it. So if you want to do it this week, if you're confident with it, and drop it in the Dropbox in our Moodle section on homework, that'd be great. And, you know, if you need a little extra time like the weekend and want to drop it in over the weekend, that's great. I just want to give you feedback, make sure you understand how to do it before we have the quiz, which again is not for a while. But again, we will do review of this stuff on Wednesday. So no sweat if you don't want to get to it right away. Okay? All right, and Mary Aldred will be reaching out to the Monday Lab with what you guys are doing this week. My group, I'd like for us to meet, to convene very briefly on Wednesday to go over a few things. And then I'll let you guys probably watch a couple videos on your own, okay? Just to have a sense of what it is that we're doing. It's Forest Community Lab, so very timely. Unfortunate that I can't drag you guys out into the woods. Anyway, thanks for hanging in. Have a good Monday, you guys. I'll talk to you later.
Thank you.
One last. That's me.
I saw a
Hi,
black
Paul.
squirrel in my backyard.
What was that?
I saw a black squirrel in my backyard.
Oh my goodness. Get on Squirrel Mapper and log it. iNaturalist. I love you guys.
Yeah, I didn't get a good picture.
Miss you.
Bye.
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 See ya.